Good evening to all the participants. Welcome to the day 13 session on ML and ML approaches of drug and vaccine design in internship and certificate course in AI and ML approaches of drug and vaccine design. So before proceeding to today's session topics, can we recap the last session topics? So what are the last session topics? What exactly did we discuss in the last session? I think each and every session we are seeing numerous voluminous, isn't it? Because the participants asking to provide more demo, based on the feedback, I'm providing more demo, but I'm speeding up. So if you want to learn, you can watch the recorded videos, pass down, and you can learn step by step. Unless you practice, you can't remember everything. Very good. We discussed something on B cell epitope based vaccine design. Especially, we have two types of B cell epitope. One is linear sequence, and other is discontinuous sequence. So, actually, one is related to the sequence based epitope prediction, other is the structure based epitope prediction. And then we discussed various prediction tools in IEDB. Other than whatever we discussed in B cell, what are the other tools and databases present? We have explored on that. And what else did we discuss? Did we discuss any other web servers other than IEDB for B cell epitope prediction? What are the tools you remember for B cell epitope? And other than B cell epitope, what else did we discuss? What are the various tools and databases we have discussed on yesterday? You are limiting your answers. Peptide, very good. So that is one of the algorithm for B cell epitope prediction. Especially we can predict linear sequence based epitope from peptide 2.0. Good. We also discussed something called pepfold3 server, which is used to design the 3D structure of the peptide. Okay, so whatever sequence we predicted, if you want to predict a 3D structure, we have to use this. And we have discussed about different vaccine databases. Very good. This is what I am anticipated from your side. We have discussed something called Violinet. Do you remember violinet.org? Here we can see human vaccines, veterinary vaccines. We can see other kind of database tools, blast and we see the Virmugen for finding the virulent genes for live attenuated viruses. And then we have discussed computational vaccine design. So actually, reverse vaccinology is one subject in computational vaccinology. So like this, if you see what are the other vaccine design, so we have discussed something called how to model the antibodies. So that itself is a separate thing, like engineering antibodies, and after engineering modeling the antibodies. Okay, for this we have seen so many commercial tools. Like in Discovery Studio, we have antibody modeler, and similarly, DNA Star is one of the example. Okay, DNA Star is also the commercial tool for antibody modeling, and also we have seen for scroid injure. Very good. We have discussed something on antibody protein docking. Okay, that is antibody antigen docking. That's what PPI. Okay, protein docking. So for this, we have seen examples like downloading a single PDB file that is hemagglutinin, uh, viral hemagglutinin talked with the human antibody. So if you try to download this structure, we can separate these two structures by editing the coordinates in the notepad. So whatever you have two different PDB protein files, we can able to dock it using the protein docking. And also, yes, very good. In scroid injury also we have discussed, we have a separate antibody modeling. So other than antibody modeling and reverse vaccinology, what else did we discuss in computational vaccine design? What are the other areas in computational vaccine design other than these two? Do you remember codon optimization? So codon optimization is one of the thing. And what else? Like if you see transmembrane helices, you know, modeling the normal protein molecules is entirely different from modeling the antibodies as well as transmembrane proteins. Okay, like especially if you focus on modeling the GPCRs, G protein coupled receptors, we need a separate servers. Okay, whatever we use the homology modeling through Swiss modeler, threading to iTaser, this won't precisely give you the structure. You find problems, okay, with the structures, even if you predict. So we have to use an exclusive servers. So we use a separate servers for the homology modeling of GPCRs. We also have separate tools for that. And also we have discussed homology modeling is one of the vaccine design method for modeling the target PDB files. Okay. Then what else did we discuss? Codon optimization, homology modeling. So these are the major things we have discussed, isn't it? Other than this, protein talking. Okay, now let me go ahead with today's session topics. Very good interaction on today. We have only one session on tomorrow. Try to listen these classes and also can interact on tomorrow. I anticipate these kind of an interaction on the last class that is on tomorrow. Very good. Now let me go ahead with this chapter 13, that is day 13 session on A and ML approaches. So before that, I just an information that I am a recognized PhD supervisor of Anna University. So myself, this is my supervisor number, and the area of specialization is this drug design, vaccine design, pharmacokinetics, and immunotechnology. And these are the areas I am guiding at present. I'm guiding for the anti-cancer drug discovery as well as anti-diabetic drug discovery. Other than that, we are also developing the drugs through the company, the newly established startup, Phyto Nanos Drug Technologies Private Limited. Other than this, I am advisory board for more than 4 to 5 universities. Okay, for many scholars, more than 10 scholars. Now let me go ahead with the today's contents. Today we'll be discussing on machine learning approach in drug discovery, and we'll be discussing something about the MATLAB, a tool for drug discovery, which uses the machine learning, and the AI in drug discovery, list of AI tools and databases in drug discovery, Python coding, programming. We see some 3 case studies related to Python coding in drug design applications. Now, drug design as well as vaccine design applications. Okay, so my students have worked related to that. I show you some kind of an outputs related to that, and related to this machine learning. So machine learning is a field of study in artificial intelligence. Okay, so machine learning is a part of artificial intelligence, and if you see deep learning, this is also a part of machine learning. Of course, it comes under artificial intelligence. Okay, so what is it majorly concerned on? It is majorly concerned on the development and study of the statistical algorithms that can learn from data and generalize to unseen data, and thus perform tasks without explicit instructions. So whatever task we perform, so without explicit instructions, we can able to process the data. So to know more about this, go to the next slide. So first we discuss about the father of machine learning. The father of machine learning is Arthur Lee Samuel. He is an American pioneer, and he is expert in the field of computer gaming and AI. So when the computer was discovered by Charles Babbage, okay, so immediately within few years, even the machine learning came, okay, by the Arthur Samuel. Now, related to machine learning algorithm, the machine learning algorithms are classified as supervised algorithm, unsupervised learning, and semi-supervised learning, and fourth is the reinforcement learning. In supervised learning, basically we can classify like classification and regression. So whatever you find in QACR, quantitative structure activity relationship studies, one of the drug design method. So here we have these two types of methods to predict the activity. Okay, so the same kind of a thing, whatever we predict in QSR is a kind of a supervised learning. Okay, and you can see these are the methods we use. These are the algorithms: linear regression, logistical regression, random forest, support vector machines, neural networks, decision trees. So most of the important things where you find the data is trained in the stratified manner, and whatever data is trained, so from the trained data set, the test data set value can be predicted. So if you want to make a good precision accuracy with the data, if you want to develop a good precision and accuracy with the data, we have to increase the population of the data. If increase the population, obviously this too will be increased, obviously. Okay, so this is what we do, and we train the data in the strategic manner. But in unsupervised learning, we use the learning task like clustering and dimensionality reduction. The examples of k-means clustering, t-distributed stochastic neighbor embedding, principal component analysis. Okay, so this is something related to unsupervised learning. Okay, and the other methods also shown below. Semi-supervised learning. This is actually in between the supervised and unsupervised. So either can have the supervised or unsupervised. So we call this as semi-supervised. And reinforcement learning can be model free or model based. These are the examples: Q learning, temporal difference, Monte Carlo tree search. This Monte Carlo you can find in simulation also, isn't it? So this exactly comes under reinforcement learning. Now related to the machine learning in drug discovery. In machine learning can be used in various applications in drug discovery, starting from finding materials, consumables in drug development, and chemical synthesis. If you have the thousands of chemicals or thousands of peptides, if you want to screen the best peptide, okay, of course, we need for chemical synthesis, we need a combinatorial chemistry, and for peptides, we can also use medicinal chemistry. So these kind of a chemistry we can use to synthesize numerous compounds. Whatever we synthesize in wet lab, the same thing we can reduce the price, okay, by just checking it using the computational tools. Okay, so this kind of a synthesis also we can make it like using the fragment based drug design. If you want to construct so many synthetic drugs from a lead compound, okay, if you want to develop so many lead compounds from heat, okay, then we can use this fragment based drug design. Of course, we can use this kind of a chemical synthesis or tagging one inactive drug with a carrier. So what inactive drug can be tagged with a carrier to make in the form of a prodrug. This is what we call it as a prodrug. Yeah, on the site of action, the prodrug is going to release the carrier and behaves like an active drug. So all this kind of a chemical synthesis, not just the fragment-based drug design, how to go ahead with the construction of prodrugs, and how to go with the cedarophore kind of a drugs. Cedarophore. You heard about the cedarophores? Actually, cedarophores are ion chelators. The best example, is the Pseudomonas aeruginosa. If you want to treat Pseudomonas aeruginosa infection, which is commonly infecting the lungs, okay, this is a lung disease. So the lungs actually, if you see this Pseudomonas aeruginosa, this can consume much of the iron in our hemoglobin, isn't it? Which protein has more iron? Only the hemoglobin. Whatever I tell, hemoglobin is a blood protein. So from blood, if iron is consuming by the Pseudomonas aeruginosa in the lungs, obviously we reduce the immunity. We lose the immunity. If we lose the immunity, then obviously we are more prone to any side of, I mean, to even up to the fatality. It leads up to the fatality. So to reduce this, okay, so we can tag iron molecules, okay, with the drug. So obviously, whatever the Pseudomonas aeruginosa can consume, can take the iron. Obviously, the drug is also taken inside. So it will be like a Trojan horse strategy. We use such a kind of a Trojan horse strategy to deceive the Pseudomonas aeruginosa. okay, to treat from this infection. So, like even to develop these kind of a siderophores, or even if you come across synthesizing the mitochondrial targeting drugs, obviously we need the machine learning. So, mitochondrial targeting regions are already available in the database. Only thing is that whatever drug we want to encapsulate or we want to tag with it, we can able to make using the linkers and spaces. For this also we can use the machine learning. Lot of applications are there in drug discovery. So drug discovery also linked with drug delivery. So related to drug delivery also we can use. But the major applications of machine learning in drug discovery in the world, the major application lies in the drug repurposing. Even for the discovery of SARS-CoV-2 drugs, what happened at that time? They used the repurposing techniques, isn't it? The clinicians, okay, in 2019, when the world is declared as pandemic in March to April 2019, what happened? The clinicians observed that there is no proper drug for this disease, so they treated using the antiviral drugs. What type of antiviral drugs are subscribed or prescribed by the doctors, clinicians at that time? Anyone know that? They prescribed 8 drugs in many regions, okay, for treating SARS-CoV-2 infection. In the earlier times, that is in 2019, they prescribed these 8 drugs. So, what are the drugs they prescribed? Anti-malaria drug, anti-dengue, anti-flu. This is okay, somewhat okay, but still all these drugs won't cure the infection. One more thing, what they have given? Anti-HIV. Do you believe even this kind of a drug is also given along with the 8 drugs for the treatment of SARS-CoV-2 infection? Then what it does? So whatever drugs that it is taken, not even a single drug is going to target the spike glycoprotein or important proteins of SARS-CoV-2. So even if you target the polymerases and proteases, how we can target these are non-structural proteins? Unless you target the core, that is a spike glycoprotein present in the structural protein, we can't able to target even the proteases and polymerases. And even that too, this spike glycoprotein is going to target what protein of human? AC2 of human. This is the major interacting thing. So this is what is giving us infection. So the target was not established. So they are trying to use the repurposing drugs for that. But later they came to know that out of these 8 drugs, they have also given one drug called remdesivir. Okay, this is actually a kind of a protease or polymerase inhibitor. So this is a only FDA approved drug, only FDA approved drug for SARS-CoV-2. Of course, this kind of a drug is also discovered from drug repurposing. That is using the antiviral drugs for a new upcoming viruses. Okay, so here also can see many results related to the machine learning. Other than this, you can see pharmacovigilance, clinical development, biomarker discovery, virtual lab. So I think you know about this. We have discussed in the early sessions. Applications of machine learning in drug discovery. So we can see supervised learning. That is, we can have classification or regression analysis. Under classification, we have natural language processing, kernel methods, natural language processing, Bayesian classifier, support vector machines, nearest neighbor. And in regression, we have linear regression and multiple regression. So along with it, we can find random forest, decision trees, etc. And in clustering methods, which comes under unsupervised, hidden Markov model, hierarchical clustering, k-means, neural networks, this comes under clustering. Neural networks also comes under in both the classification as well as in the clustering. A basic machine learning system rearrangement. So how we start populating in the training data and how to use the training data using the machine learning algorithm, and we come up with the model evaluation finally. So whatever algorithm we use, if you want to check the precision accuracy, like how exactly it is going to produce the results. Obviously, we use a kind of a workflow, isn't it? This is the workflow we use. This is taken from this article. On commonly used machine learning software tools, we have plenty of tools. The most important tools what we use is of course Python programming, no doubt in that. Like in Python programming, you can find SA, Kitlearn, Brain, and for the cloud computing, if you have a large amount of data, large data, big data, we can try with this TensorFlow. Okay, and R programming, popularly used programming, okay, in biotechnology application, especially in bioinformatics applications. This also comes under this machine learning. And VK, my supervisor, Professor Michael Grumiha, who is number one bioinformatician, he is using this kind of a tool developed in Java, VK, and MATLAB. It's a multi-paradigm programming language. So these are coming as you can use it as a machine learning tools. And commonly used machine learning tools we have discussed. Now we are discussing about the practice and applications in AI. Anyhow, about the AI we'll be discussing later. Okay, these slides you can see later. So one such example of breast cancer prediction in machine learning classifiers. Okay, we can use the support vector machine, logistic regression, k nearest neighbor, ensemble classifier, etc. for the types of breast cancers. We have breast cancer caused by different genes, and even we find triple negative breast cancer, which is resistance to three important genes. So related to that also, we can use the kind of a machine learning algorithms. You can use even multiple machine learning algorithms to check which is the best model we can predict from that. And whatever we discuss, the computational methods in drug design, the tools like MolSoft, Mol Inspiration, Swissadium, everything are developed only to the machine learning technique. No doubt in that. Already these tools are developed. But what are the gaps present? For what applications we can use the machine learning applications? That one day I am discussing from the beginning. Okay. So drug repurposing. Okay, of course, drug repurposing for any specific disease. If something like a SARS-CoV-3 is going to come in future, or even in SARS-CoV-2, we have hundreds of strains. We don't have an approved FDA approved drug for all these strains. For general strain, we have a remdesivir, that is the only FDA approved drug. How about the other strains? Even if you want to come up by improvement with remdesivir, what else we can bring? Okay, so everything can try with this drug repurposing, of course, and any ML approaches of prediction of repurposed drug. That's what I was mentioning you. We can see around 380 entries of SARS and 342 entries related to SARS-CoV-2. We also have 401 entries for MERS. So why can't we develop specifically for few entries using this machine learning techniques? We use a combination of machine learning techniques also, try to build a good model, and through the model we can able to evaluate. How to evaluate? Obviously use docking, simulation, etc. whatever we predict. Bring in new candidates also using fragment based screening and try to use the docking and simulation to check the binding and stability of the drugs. Now some applications of machine learning techniques, okay, with respect to the tools whatever we are using at present. Swiss ADME. Do you remember this diagram? What is this we called? Boiled egg diagram. So what is the purpose of boiled egg diagram? To check whether your drug is a lipophilic or hydrophilic, we use this. Just uploading a smiles format, we can able to see after executing it, we can see the drug present in boiled egg. If the drug is present in the white portion, this is actually, I told you, this is an albumin. You find much of the proteins in this white region and much of the fat. Okay, this is what a yolk we find in the egg, isn't it? Much of the fat you can find in the yellow part. So if our drug is present in the yellow part, obviously it is a lipophilic drug, but here it is present. So it is an hydrophilic drug. This is how we classify the drug is a hydrophilic or lipophilic. And however we classify, not only we can check with a single drug, we can check with numerous drugs. If I have nearly 500 drugs, and if I want to check the nature of the drugs, okay, I want to screen the lipophilic versus hydrophilic, I can use SVM. Each and every dot is a drug. If you see, whatever this line I get, okay, I have this two lines, blue lines, isn't it? So above the blue lines, I mean above these lines, we can see so many red dots. It means that we can able to segregate. Lipophilic drug is shown in red color. We can able to separate the lipophilic drug versus the hydrophilic drugs using the support vector machine. The support vector machine uses a vector to separate these kind of drugs. Of course, we use a kind of a hyperplane. We call this as a hyperplane, a line that separates the data input nodes with different values. Got it? This is an example. SVM is default used in Swiss area. Now let me come across with deep screening. That is a deep learning technique. So where we are using this deep neural networks. Okay. So this deep screening, we are using even in QSR prediction, quantitative structure activity relationship. Any PER, QSPR, any kind of a properties like activity, toxicity, anything we can predict using this deep learning techniques. Okay, where the deep neural networks are used. Of course, this is also used for analyzing medical images in various fields. Like if you want to diagnose the stage of cancer, or you want to diagnose on which day the SARS-CoV-2 is infection we have. So for this also we can use this deep learning technology. The next is the K nearest neighbors. You heard about this? Just now I explained you. This comes under unsupervised learning. This unsupervised learning you can able to see in this Jing 15 or Jing 20 tranche databases. What is this actually, Jing 15 or Jing 20? This is a synthetic molecule databases. In the synthetic molecule databases, we have molecular weight versus log P. Okay, that is, if you see the molecular weight, we have molecular weight from 200 to 500, and even above 500, we have a set of molecules. So we can see the molecular weight is in x-axis, and in y-axis we have partition coefficient. If I want to screen a log partition coefficient of 3 and the molecular weight of 350, whatever I select these two, I'll be getting this much number of molecules in the database. Got it? 350 molecular weight mapping with log P3 is mapping to 37, around 37 lakhs. Huh? So this much number of molecules, okay, 370 lakh molecules. All these molecules, I can download it and it can use it. Okay, so here how this is mapped. Okay, this kind of a mapping is what K nearest neighbor. Okay, so this works near the K nearest, of course, unsupervised learning. Now, random decision forest, of course, a part of a supervised learning. Here, the best example is how a tool Galaxy QSR is developed. That is, you can develop the training data sets, and then from the training data set, value can be predicted. That is, for a test data set, value can be predicted from the training data set. So the QSR application, you can able to see that can also be done through random decision forest. And we can also see one more application, machine learning technique from the neural networks. Okay, so if you see in the neural networks, so we can see this is the most important applications. And even this kind of an application I have published in an Indian patent. I also have an Indian patent related to this. So how it works is see this thing. So we have to get initially a lot of X-ray images. What is this actually? The X-ray images to detect the SARS-CoV-2 infection. If we have, if the patients have more SARS-CoV-2 infection, so the black gray spots are not visible. See here, it is white here. If we find a white color here, obviously it means that the patient is having 100% infection in the lungs. That is, he is in the day 7 or day 8 of infection. There is something called quarantine period and infection. So this is actually an infection period around that 7 to 8 days period. Okay. So like this, if you see whatever X-ray images, we can use the heat maps in the tool. In the software, we can use the machine learning tool. We have a heat maps. So wherever you can find this red and yellow, see this. This is aggressive infection. This is also aggressive infection. Okay, so that is in the 7th and 8th. Like however you find in the X-ray images, these are shown like this in heat maps. So when compared to this white, black and gray colors, we can also see in these colors, red and yellow. Got it? So like this, you can be able to use the machine learning techniques to generate the heat maps for diagnosis. What is this for? This is for diagnosis of viral infections or even for the cancer diseases. And the software we use is the NIMS, TensorFlow, VCAR, RapidMiner. Now let me go ahead with the next thing, R programming. So R programming, how the R programming looks like for virtual screening. We remember virtual screening, what we have tried with PyRx, Python RX programming. The name itself is Python RX programming. So here we are developing a kind of a tool. Okay, we can able to develop any kind of a virtual screening tool. Okay, for the developing a tool using the R programming, this I have taken from the Adrian Journal of Cheminformatics. Okay, so we have to include the library molecules. Like in, for example, if you want to use printf and scanf in C language, we use stdio.header file, stdio.h, isn't it? Similarly, if you want to screen the drug candidates based on physicochemical properties, we have to use the functions from the libraries. So to include the package libraries, we have to use the code like this. Okay, so RCT, chemometrics, something related to this gives you some knowledge on 1D format, SMILES format. So to take the SMILES format, to take the 3D structure, SDF formats, the chemometric library has the functions related to that. Okay, and RJava, chemomine R. So for mining the data, we use the functions from chemmine R. So for clustering the data, we use the cluster library. That is the package we are including to this library. So for using these packages, of course, we can install the packages in the R Studio. After installation only, we have to use this kind of a code. And using a kind of an algorithm for screening based on physicochemical properties, we can able to screen with our own software with the machine learning technique we have deployed. And now let us come to the MATLAB machine learning. So if you see the MATLAB machine learning, okay, so whatever we discuss, supervised and unsupervised, we can also see the different machine learning algorithms, sub-classification of algorithms, whatever we have studied, whatever we have seen in the previous slides, we can see everything in the MATLAB itself. Like Bayes method is there. What for Bayes method is used? The Bayes method is best used to construct these kind of networks, PPI networks. So having connecting multiple nodes, the Bayes is the best algorithm used to construct the networks. Nearest neighbor, just now we have seen related to tranches, like downloading the data sets from downloading the multiple molecules. So even not only the machine learning techniques are used to develop the tools, also used to construct the databases also. Okay, so of course from databases only we use the tools for mining the data. For mining, we use an exclusive packages. Okay, so now let me go ahead with the MATLAB. The MATLAB can also associate with few more tools like TensorFlow, which is linked with the cloud computing. Okay, the TensorFlow is of course linked with the Python programming. We have a separate programming for MATLAB, but we can link that the Python programming with MATLAB. Okay, like the MATLAB is something linked with a tool called Deep Learning Toolbox, where we can integrate these two programming. Like how the HTML can be linked with scripting languages like JavaScript, VB script. Similarly, we can integrate these languages. And related to if you want to learn the MATLAB courses, we have MATLAB online courses. Fortunately, the Sri Venkateswara College of Engineering has a MATLAB license. That is, each and every student and also a faculty has the access to work on MATLAB. Okay, so if you have a MATLAB license, we can able to even complete these online courses free of charge. Like how we can learn Coursera, okay, Udemy. Similarly, we have the courses, 6 modules, 2 hours, 6 modules, 12 hours. Okay, machine learning on ramp, machine learning with MATLAB, deep learning on ramp. So like this, we have the courses where we can study in online. And machine learning using MATLAB online course. How to start the course? Just I've opened a machine learning with the MATLAB, the basic course. Okay, so these are the course modules. So if you get inside, we find this kind of information. Also, if you want to learn machine learning, I suggest you to go through this tool, this online website, w3schools.com. Very easier to use. I mean, if you learn here itself, you have the compiler, where in the compiler you can execute the code and you can learn. Okay, if you want to have so you're starting, you are in the starting phase, you use these websites to learn. Now, let me go ahead with the AI. Till now, I discussed on ML. AI, I told you it's a software, it's a major thing where you can find machine learning and deep learning coming inside, isn't it? So, what is actually an AI? It's a software program that has been trained on a set of data to perform specific tasks like recognizing certain patterns. Okay, and if you see the AI models, whatever we see in the machine learning comes under AI models. Father of AI is different from machine learning AI. I mean, father of machine learning. So that is John McCarthy. He is an American computer scientist. He uh, he is the father of AI. He played a seminal role in defining the field devoted to the development of intelligence machines. So, along with the machine learning, even this AI by John McCarthy has brought this term, has coined this term. AI, okay, AI, I told you it links with the machine learning. We can use all these machine learning techniques for drug design as well as the vaccine design, whatever we discussed earlier. And this is the slide I skipped earlier. That is AI, drug design applications in AI. So whatever we use the machine learning like target identification, hit discovery, hit to lead optimization, and whatever methods I showed earlier as computer-aided drug design, all the applications related to drug and vaccine design, of course, are developed using machine learning, which is nothing but a subset of AI. Okay, including QSCR, pharmacophore mapping, modeling, docking, virtual screening, everything is developed. Okay, so we are using the tools for automate. This is automated. If you give any kind of an inputs, obviously how we are predicting it, AI and ML is integrated inside. Applications of AI in drug discovery and development. Okay, so whatever we have seen related to clinical trial design, quality assurance, quality control, pharmaceutical manufacturing. Also, we have AI, isn't it? Like if you see in the pharmaceutical industry, how the drugs are taken, how the API is taken, how the fillers, surfactants. If you feed everything in the process control thing, we can able to see that kind of inputs are taken, and we'll be producing that drug. Okay, so temperature, pressure, humidity, all the conditions are monitored using this machine learning techniques in the process, in the pharmaceutical manufacturing, and similarly, drug discovery, and of course, development in our labs. AI for drug discovery can also be extended to biomarker development. Like if you want to find which is the best gene, I mean, which is the most frequently observed gene for a specific type of cancer or for rare diseases. So that where you find the biomarker discovery is most important. Unless you find a target, the most important target for a specific person, we can't able to tailor the medicine. We can't able to give the precise medicine. Of course, to detect from the genomics data and finding a biomarker, we can also use the AI. Okay, so we can use any of these machine learning techniques from the biological data and pharmacological data, whatever we have. And related to in silico approaches in drug repurposing. Okay, for drug repurposing, I simply mentioned whatever drugs available in the database like PubChem, Drug Bank, we can reuse for the upcoming viruses. But what exactly is there? What kind of workflow is there? See, first we can in the drug based we have chemoinformatics data, and we have to check even side effects data. For example, Drug Bank has drug-drug interaction, food-drug interaction. Of course, we have to check everything, and even we have to check the toxicity. So all these things, okay, it comes under drug based, and checking the drug likeness filters, checking the drug likeness values, checking what are the metabolites that upregulate or downregulate the expressions. Of course, metabolic data is needed. All the types of omics you can see also integrated with the data mining, gene signature, gene expression data is needed for the disease based epigenetic data, post translational modifications, and how we can link the drug with different diseases, how many drugs we can get from a single plant, everything. Okay, so about the phenotypic data, like if you have data sets, geo data sets and other kind of gene data sets, okay, how many persons are majorly infected by a particular gene. Like even in the last session, day before yesterday, I told you we have allele frequency data. From the allele frequency data, we can also target specifically a community or even a region specific for a vaccine design. Similarly, even for the drugs. Okay, so drug based, disease based, and even target based. Not just focusing a single target can able to treat a disease. We need even the targets, the client proteins interacting with the targets. What are the other client proteins? That's also important. And even how about the gene expression? How about the positive regulation, negative regulation? So we have seen in ovarian cancer, kidney cancer, journal papers, whatever I published. So these papers, okay, something related to target based, of course, linked with docking, dynamic simulation. So everything, all the things we have to use it for drug repurposing, not just downloading and just doing with the docking. All the things has to be considered properly. Okay, for utilizing it for the in vitro studies, for testing in vitro studies, or up to the in vivo, and also up to the clinical trials. Pipeline of AI-based drug discovery development of COVID-19. I told you in the SARS-CoV-2 virus, we have structural proteins and non-structural proteins. The non-structural proteins, we have main protease, chymotrypsin-like protease. We have types of proteases as non-structural proteins. And most of the viruses we have RNA-dependent RNA polymerase, RNA-independent RNA polymerase. These are also comes in a non-structural proteins. You know, whatever I told, the remdesivir is not targeting the spike glycoprotein. But if it is not targeting this is targeting these kind of proteins actually. Of course, this is a repurposing drug. Only repurposing drug and only FDA approved drug. I mean, only repurposing drug which is FDA approved. FDA approved, it means that it has cleared up to the third phase trials. The remaining thing can clear up to the phase 1 and phase 2, whatever drugs available. Okay, and you know why we the most important, if we don't have a drug, especially to target the spike like a protein, that's a problem. If you have a drug, then obviously, whatever the most important protein interacting with AC2, this can be, uh, this kind of interaction we can inhibit. Okay. So for this also we can use the ANML and we can bring with few more drugs from fragment-based screening or other based drug delivery-based screening methods. Computational drug repurposing. So we use the networks, we use the virtual screening, use the homology modeling simulation, everything. Okay, for drug repurposing. I told you we have to use almost all the techniques, all the omics to find the exact thing. Like high throughput screening can be done through docking, pharmacophore modeling, genomic we can see. Okay, so these are some of the drugs discovered for the treatment of various diseases. Okay, repurposing drugs, and we have seen this also earlier. Now let me go to the main application, AlphaFold. The AlphaFold is an AI enabled protein structure database. So AlphaFold is also available in RCSP PDB. Other than this, we also have an exclusive website URL to download the protein structures which are modeled through AI technique. Okay, so around 200 million protein structures are available because of this AI. Or else we have to use just 13 lakh structures. That means we have just 1.3 million structures available at present. Just because of AI, we can able to see this much population. Now related to list of AI softwares. So if you want to use the AI related softwares for various bioinformatics applications, for drug design applications, we can use this URL bioinf.jku. So like this, we have plenty of URLs to use the AI tools, and the most I suggest you to use these kind of tools, that is AI tools which are developed in protein bioinformatics lab in IIT Madras, Chennai. So this was developed by group Professor Gromiha. So he has developed so many kind of tools. Okay, for example, the tools developed in using the machine learning, each and every tool is a product of a PhD scholar. Is the PhD thesis. You can find for predicting the binding affinity of membrane protein complexes. For protein docking, we can be able to use hex dock, patch dock, etc. How about membrane? That's what I told. The membrane proteins are something different. They have a complex structure. For example, if you have the membrane proteins, especially the GPCR, okay, if this is your cytoplasm and this is an exterior part, okay, of your membrane, this is an exterior, this is an interior. Whatever that GPCR, okay, this protein can go inside and outside of the exterior and interior part of the membrane. So obviously, modeling these kind and predicting the binding affinity, even whatever drugs we predict for these drugs, predicting the binding affinity is more difficult, isn't it? So he has developed a kind of a tool called MPA-PRED. Okay, and we can also see for codon de-optimization. Do you remember? This is also a part of a computational vaccine design. Codon de-optimization. And similarly, proton DNA complexes binding affinity. Similarly, we can also see for carbohydrate protein binding, lipid protein binding. That's also available. We have also developed for identification of protein potential driver mutations in glioblastoma using machine learning. Like this, we see plenty of applications, whatever is not available in the global web servers. Okay, so more than 20 tools and 20 databases are available. So we can also see some databases related to the coronavirus related antibodies. Okay, we can able to see a curated database for binding affinity and neutralization profiles of coronavirus related antibodies for this ABCOV. Like this, we have plenty of things related to the mutations, related to protein aggregations, related to membrane protein thermodynamics. Okay, it's a challenging task. Whatever is there, already tools and databases are available. And myself, I also have one Indian patent related to biomedical imaging system for cancer screening using AI. Okay, I'm one of the author. So developed along with IT and CSE faculties, scientists. So different claims. These are the six different claims. Whatever claims, whatever I mentioned earlier in detection of lung cancer using the X-ray images, using the microscopic samples. So all these things I have used under my claim. Okay, using the microscopic samples, even we can distinguish the types of cells, etc. Isn't it? Using the phenotype, that is the role I've used in my claim. And role of AI in drug discovery. This we already seen in multiple slides, isn't it? And AI in cancer research. So AI in cancer research, we can use it for cancer prediction. It includes cancer incidence, cancer recurrence, and cancer survival. So in the cancer, actually, the AI can be used for prognosis. That is in the earlier stage, stage zero itself, we can able to find, and even we can use it to find that for diagnosis. And the AI can also be used for treatment of cancer. For all the applications we are related to cancer, we can use AI. Of course, here for the diagnosis, we can use sensor-based detection, biomarker detection, self-diagnosis applications, like this. Okay, and even in the cancer treatment, you can see these are the applications. Whatever we do, cell programming, like identifying the cell and checking in which stage through the phenotype, and cancer earlier diagnosis. Okay, so from prognosis to treatment, we can use it. Biomedical imaging, that is up to the microscope. If you take, we can, so like for example, if you want to detect the bacterial infection, you have a positive staining or negative staining. So these colors can also be identified multiple slides if you have thousands of slides. So these slides we can able to trap in the form of images, and we can also infer through statistical inference by measuring the number of cells and how much it is stained, and for each and every day after treating the drug, we can able to make it. Like in the big hospitals, okay, if a patient is diagnosed with any kind of bacterial or viral infection which is very severe, okay, then obviously we can go with this kind of biomedical imaging through the AI techniques. Of course, SVM classifier can distinguish the types of the positive staining, the positive and from the negative staining. Illustrative figures, like earlier, whatever we tried with the breast cancer, similarly, the AI can also be used for lung cancer. I told you, like, however, we can use the X-ray images for detecting the SARS-CoV-2 infection. Here we can able to detect related to the stages of cancer. There we can detect up to the day of infection. Okay, so like this, you can see in multiple applications. This is related to breast cancer. This is related to the head and neck cancer. What are the different morphologies? And AI for patient stratification. So generally, the AI is used to personalize the medicine, to come up with a precision medicine. Nowadays, instead of relying on a single scientist with a single publications, we can also have a tools and databases or existing tools and databases. The only thing is that we have to mine the data, and we have to certain to is treatment of a specific disease. Okay, all these things we can able to bring. Okay, and if no, if we find the gaps, if we don't find a specific tools, we can develop the tools for the data available in the databases. So this is actually an adjuvant immunological database. This I have developed, but this is actually I just use these kind of a programming. We can't tell that exactly it is a machine learning, but we have very less time. So this is actually case study one. This is what my students have done. I told you I will be showing you three case studies. Case study one. This is the Python code for toxicity feature prediction and ROC analysis. Okay, so this is a latest done project by a student called Nitisha. So actually, she has determined a receiver operating characteristic. okay, for uh, the hepatitis E virus infections. Okay, so we can get four types of data, true positive, false negative, false positive, and true negative. Then under this, for checking accuracy and the precision, we can also use this receiver operatic characteristic, okay, which can determine the type of data actually. So this is a Python code for toxicity feature prediction she has used. Of course, whatever the results from Protox 3, we have predicted using through this programming, and this is the model we have generated. Okay, and we can able to find this random forest AUC as 0.92. This is the best algorithm we have predicted. And related to case study 2, heat map analysis of docking scores from Python programming. We can use the Python programming inside before using the Python programming. We we have to install the packages. Like however we install the libraries in the R Studio, even in the Python programming, we can use the editors. Okay, we can use the Galaxy editors, and we can able to install the packages, and through the packages we can use the functions. Okay, so we can install multiple libraries. I'm showing you after installation how you get these kind of an output in that uh, notebook, Python notebook, and Python programming of docs course. Okay, so this is the code we have used, and from the docs course, multiple docs course, that is docking multiple ligands with the multiple targets. So I'm using a kind of a heat map. This is a kind of an output which generated from Python programming. Okay, heat map images. And case study 3. This actually I am predicting the accuracy, the precision, AUC, recall from logistic regression through Python programming. Okay, so whatever we use this Python programming, okay, we are using this programming for uh, training data in the data sets, okay, and whatever training data sets we have, we can also predict using the test data set. So for this, we tried with hepatitis. Okay, we tried with hepatitis. But earlier, whatever I showed related to Nitisha, 2025, she worked on breast cancer. The case study 1 is related to breast cancer, and case study 2 is related to hepatocellular carcinoma. Case study 3 is related to hepatitis E virus infection. Here also we use the same kind of Python programming. After installation of Python libraries, we have to use the code, and we can use the functions from the packages, whatever we have imported. So using this code, okay, the Tamana, she is the PG student who worked under me, and she worked on different kind of uh, the diseases linked with the hepatitis C. Like if a patient is suffering from asthma, and that also that patient also is presently having the hepatitis C virus infection, how about the precision of coming up with a kind of a treatment? Okay, so for this, we are using this kind of a machine learning actually. So using this machine learning technique, she has used a linear regression method, and using the linear regression method, okay, she came up. I mean, these are all the codes actually she has used, and we have used a lot of kind of factors inside. And after developing a training data set, this is the final output we have received. That is, whatever data we have in the data sets in the databases, okay, after using the Python programming code, we determine the accuracy of logistic regression. Like this, we can try with multiple machine learning techniques. The accuracy is 83.3, and AAC we got like this, recall. So how we can improve the accuracy, precision, etc. If we increase the data, then obviously we can increase the relevant data, can increase the accuracy and the precision, so that it eliminates the other three false data. The true positive alone is accepted. This is not accepted, this is not accepted, this is not accepted. So if you get a related data, we can increase the true positive, and obviously the accuracy and precision will be more. Okay, so I have completed today about the AI and ML applications of drug and vaccine design. Okay, any doubts you can ask me. The only thing is that you should have a knowledge on Python programming. So R programming, I think many of them are trying for it. If not, the R programming is easier to practice, but it is limited to certain applications. So if you want to use much of the applications related to prognosis, diagnosis, treatment, obviously I prefer Python programming because we have plenty of packages to use the functions related to this, whatever I mentioned. Okay. Any doubts at present, you can ask me. We have only one session at present, that is on tomorrow, to complete this internship and certificate course. Any doubts? Can be any doubts, you can ask me. So, want to learn program? What should I do, sir? Actually, even Python programming, I suggest you to use even w3schools.com. w3schools.com. You remember I showed you in that video in this session. So you can use that to learn the basic skills. And for MATLAB programming, we can learn from the MATLAB online tools. But one thing we should have is that commercial tool to practice. And in the computer-aided drug design, okay, I just also included a special module like providing a demo on MATLAB, how to work ADME pharmacokinetics using MATLAB. Okay, and if you want to learn programming, even myself, our company, Phyton Nanostruct Technologies, is conducting one course on which starts at 21st of January. That is disease detection using AI and ML. There, in this, I have recruited two course instructors. One course instructor, Dr. Vijayanand, he is actually an ex HOD of a reputed engineering college. He is a pioneer in AI and ML. He already has received grants related to disease detection, that is, which is linked with the Python programming. Okay, and another course instructor name is Mahalakshmi. She is from different college, from Ramakrishna Engineering College. She has completed PDF and PhD in ANML, especially in disease detection. She also has her own startup related to this. So I just using the pioneers for training in this Python programming. Like however I'm training, even these scientists are also going to train. So which will this course will be commencing on 21st. So you can learn parallelly that free skills from w3schools.com, and even if you want to use it for any kind of these applications, biological applications, you can also enroll to our course. Okay, obviously you can learn something more other than whatever I'm teaching. Here I'm just teaching you only with the theoretical things, and I showed you some kind of a projects whatever I completed. And of course, I for this purpose only I'm taking one course. This is the first time I am taking a course related to Python programming, that is on tomorrow. Okay, so you can also learn some fundamentals of Python programming. What exactly is Python programming? How to use it? How to install to work with the Python programming? All these things, some basics I'll be teaching on tomorrow. But more applications, you can also learn from the upcoming courses, whatever we are conducting from Phytonanos. You can also spread to your faculty members or colleagues, even or your scholars who can work under these kind of specialization. Okay. So any other doubts, you can ask me. Okay. If any doubts you have, you can accumulate it and you can ask me on tomorrow. So last 5 or 10 minutes, we just have to discuss to clarify the doubts from one session, first session to the 14th session. So any doubts you can ask me on tomorrow also. Okay. So thank you very much, participants, and I really feel blessed because I got so much good interaction on today. Okay. I too got impressed on your interaction. So similar kind of interaction, or even you can improve your interaction on tomorrow when we recap the last session topic. So we have so many proteins are responsible. How to select out of one out of it, or do we need to work in all? Actually, we have to focus only on one target. For any research, we have to target only one protein. Okay, well, but we can try with the interacting proteins. Or if you are trying with multiple proteins, we have to screen which is the best protein. I mean, the best protein we have to check only through the frequency of occurrence. So, what is the best target? I mean, what is the frequently observed target protein for a specific cancer based on the data sets available in the database? Like this, we have to do the analysis and then we have to show this. Okay, something like that. If your target in the drug design, I told you we have target deconvolution and target discovery. So trying with multiple targets or trying with a single target, it's just the application in where exactly we want to fit. Okay, that matters. Okay, so if and if you step in research, you learn a lot. So learn this kind of basic skills and try to. And also I guide the students, the external college students in the form of consultancies. The students can come to the SVC. The scholars also can. We can also arrange accommodation, so especially for boys and etc. We can arrange the accommodation, and they can. I know many students stayed here. They work for 2 or 3 months and they learned so many skills. They completed the projects. Now we are going to publish the publications for them. Okay, so everything is feasible. Okay, bringing up to the outcome of publication is feasible through guiding projects in the form of consultancies. Okay, so you're welcome to even write funding projects under thing. So even if you want to put me as a mentor or any kind of a guide, okay, for any kind of funding projects, whatever, right? Even you are welcome. Okay. So thank you very much, and just follow my kind of a LinkedIn profile, and use my WhatsApp mobile number to keep in touch with us. Okay. Have a great day to you all. Bye.